So now we'll discuss about the isomerism in amino acids. So isomers are generally classified into structural isomers and also the stereoisomers. So the structural isomers are namely the isomers which are having the molecular formula which is same. So they are having the same molecular formula but have different structure. And because they are having different structure, they are called as the structural isomers. So it is generally made up of chain isomerism, position isomerism, and also the functional isomerism. So we are not concentrating on the constitutional isomers right now. We are majorly focusing on the stereoisomers, which are also the spatial isomers. So in stereoisomers, we have generally the geometrical isomerism, which is focusing on the geometry, and the next one is the optical isomerism. So optical refers to the light. So in optical isomerism first, we'll discuss about the enantiomers. So enantiomers are generally composed of the single bonds. So only which are having single bonds will be considered over here. And chiral center is a must. So glycine cannot come under this category because it does not have a chiral center. So it cannot form any enantiomers. It does not have any D or L forms. But we generally require the D or L form. So the capital D, capital L refers to the position of the amino group, and small d and the small l represent the rotation, whether it is rotating towards the right or the left side. And the isomers, that is enantiomers, are the mirror images of each other which are non-superimposable. So let's look at an example over here. So when we have a compound such as this CH3 OH OH C2H5, so this can be written this way: H H OH OH. So this is the first compound and this is the second compound. The only difference over here is that it is composition is same. So in the stereoisomers the composition is same, but the connectivity is a little different. So over here the connectivity is also same, but how it is connected is different. How it is connected to other atom. So I'll give you an example of this. See, if you this is actually a C which is represented by a stick, and this is also a C. So over here you see that H and OH, it is connected similarly. This one also H OH. Connection is similar, but how it is connected is different because OH is towards this side of the C, that is towards the D side, and H is towards the L side. So similarly, we see the same thing. Over here the connectivity difference is present. So these two are enantiomers, which are mirror images of each other, but you cannot superimpose them on one another. So 3 and 4 are enantiomers, and 1 and 2 are also enantiomers because these are mirror images, but you cannot superimpose them on each other. 1 and 2 enantiomers, 3 and 4 enantiomers. So how do you identify an enantiomer? It should look like a mirror image, first thing. And how do you differentiate an enantiomer from a diastereomerism? That is, over here you see that this one and this one is non-related more than this and this. So these two are more related than this and this, and hence more related form the enantiomers and less related are the diastereomerism. So this is like a layman's term. So these two are diastereomers. Over here 1 and 3 is a diastereomer, and also 1 and 4 is also a diastereomer. Hence 2 and 3 and 2 and 4 are also diastereomers. How do you identify how many stereomers a compound can form? Through this simple formula, 2N formula. So that is very simple. How many chiral carbons can you see in this compound? So 1 and 2. How? The connectivity is different. So when you see this carbon, you have one functional group, that is CH3, the other functional group is H, the third functional group is OH, and the fourth functional group is the total whatever is present over here. So it's different. And when you look at this chiral center, you see OH is one functional group, second one is hydrogen, third one is C2H5, and the fourth one is this one. So they are all connected to different functional groups. But when you look at this carbon, this is having all three hydrogens. So this is not chiral carbon. And this is C2H5. This is also not chiral carbon. Because if you look at this will go this way. C, C, 2C and H, 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 H. So H, 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 H. So this carbon is connected to 2 hydrogens. So this is not chiral. And this carbon is connected to 3 hydrogens. So this is also not chiral. So this C2H5 is also not chiral. So only the second and the third carbon are chiral. So because we have 2 chiral carbons, you have this 2 as common, and N refers to number of chiral carbons. So we have 2 chiral carbons. So when you put 2 par 2, you get 2 into 2, that is 4. So in total you get a 4 stereoisomers. So hence you got 4. 1, 2, 3 and 4. So that is how you calculate. Suppose there are 3 chiral carbons. So you put it this way, 2 par 3, that is 2 into 2 into 2. So this is 4, 4 2s are 8. So total you get 8 stereoisomers. So that is how it works. And the second one, so we have talked about the enantiomers and the diastereomers. Now we have to talk about the geometrical isomers. Geometrical isomers are present in 2 forms, that is cis form and the trans form. So I'll give you an example in this structure itself. So when you look over here, OH is present on one side. So this makes it cis. If they are present, functional groups are present on the same side, this is called as cis. But if they are present on the opposite side, that is in case of the second structure, you see that OH is over here and OH is over here. So this is in the trans configuration. So this is called as trans. So now we have learned all the basic properties of an amino acid. So let's now look at the amino acid structures. So we have seen that there is an alpha carbon which is connected to an carboxylic group, an amino group. So this is generally written in deuterionic form. That is, both of them are carrying charges. Both of them are in the ionic form. And this is connected to one hydrogen. So we know that this is the common structure. A C alpha carbon which is connected to a carboxylic group, which is connected to an amino group, a hydrogen atom, and the R functional group. So we are now looking at the functional group. For glycine, R is H. So this is a simple carbon because it is not chiral. This is having no optical activity. Next we have alanine. Alanine is nothing but you just need to add one CH3 group. So methyl group makes it alanine instead of R. But instead of R, if you make 3CH, so you have C, C, C. This will make it valene. So this is now alpha carbon. This is the beta carbon. So branching at beta carbon makes it valene. Then you have leucine. So this is alpha carbon, alpha, beta, gamma carbon. So branching at gamma makes it leucine. This is gamma symbol. So you look at methionine. Methionine is alpha, beta, gamma. Then you have a sulfur group. Then you have another CH3, a methyl group. So methionine is a sulfur in between. You need to remember this. And isoleucine is alpha, beta. There is no branching over here. It's a straight chain. So over here in beta chain, you see that there is branching. That is, one is having C2H5 group. So this is C2H5 group. And this is CH3 group. A methyl group and an ethyl group. Because this is asymmetric branching. One is methyl and one is ethyl. So over here, this first group, which is consisting of the 6 amino acids, all of the 6 are non-polar, aliphatic. So that means these groups react with each other. So when you have a protein and when you have water outside, when these amino acids are present, they tend to aggregate. They tend to aggregate and they tend to come closer, having non-polar in between. That is, non-polar groups will be inside and the outside ones will be polar. Because non-polar hates water. That is, it is hydrophobic. It's having a phobia towards water, so it comes inside. So all of these are non-polar. So non-polar groups are present. So in the lipid bilayer. So if you consider this as the outer membrane, this as the inner membrane of a cell, you see that non-polar groups are generally present in the membrane, inside the membrane. These are also called as the in the membrane pass proteins. So now let's talk about the polar and uncharged R groups. So first in the list is the serine. So you have the alpha carbon group, then you have the beta carbon group, and then you have the hydroxyl group. So whenever in chemistry we talk about naming a compound, we see the longest chain which is possible. So this is the longest chain, right? So because in the chain it is attached directly, this is the primary hydroxyl group. But when you look at threonine, the longest chain is this. So hydroxyl becomes secondary. So this is a secondary hydroxyl group which is attached. For the threonine, we see that alpha carbon, we have a beta carbon. So there is a methyl group attached to the beta carbon, and hydroxyl group is also attached to the beta carbon. It's just an extension of serine. Then we look at the cysteine. Cysteine is having a thiol group, which is also referred to as the sulfhydryl group. Sulfhydryl group. So this is the thiol group, that is SH. So you have alpha carbon, beta carbon, and then SH group is attached to the beta carbon. Then next you have the proline. This is a special group. All other amino acids are referred to as the amino acids, and only proline is referred to as the amino acid. That is because this is having a secondary amino group. So we see that in all other cases amino group is free. We see that amino group is free everywhere, but only in proline the chain comes and again attaches itself to the amino group, making it a closed structure, and this itself gives the property of rigidity to the proline. So it's not at all flexible, but glycine is the most flexible. So there are two properties over here. One, glycine is the most flexible, and two, proline is the most rigid. So these two are the most structure destroying amino acids. So they will destroy the structure stability of beta sheets. So now if we look at proline, this is also called PYRROLIDINE, pyrrolidine. So this is the pyrrolidine structure or pyrrolidine structure, and this is heterocyclic and this is very rigid. Now let's talk about aspargine and glutamine. So aspargine and glutamine are nothing but the amide derivatives of aspartate and glutamate. So if we look at the structures of aspartate and aspargine, we see that they are quite similar. Alpha, beta and carboxylic group. So here also alpha, beta, and then there is an amide group. Over here this is C double bond O minus. Here it is C double bond NH2, an amino group. And if you look at the glutamine, same. It will have a gamma carbon extra compared to aspartate. So this is how you remember the structure. A more longer chain is glutamate and the little shorter chain, only till beta, is the aspartate. And similarly, you remember aspargine and glutamine the same way. That is alpha, beta, gamma. Instead of a carboxylic group, you have a NH2 group substituting the oxygen side. So that is how you see the polar uncharged R groups. So even though these are polar, they do not carry any charge on the R group. The next one is the negatively charged R group. So we have dealt with this also. Now that is positively charged R group. We have three amino acids in this condition: lysine, arginine and histidine. So when we look at the lysine is a basic amino acid. Lysine, arginine and histidine, all of the three are the basic amino acid. So this is alpha carbon, beta, gamma, delta and epsilon. So an amino group is attached to the epsilon group. So this is how you remember it. So there are carbons till epsilon group, and amino group is attached to the epsilon carbon. And then you have arginine. Arginine is the most basic amino acid, which is having a guanidinium group. So this is the guanidinium group. The NH, C double bond NH2 plus and NH2. And then you have the histidine. The histidine is having an imidazole group. And this is more stable in the physiological pH 6, and hence it is present in most active sites of the enzyme. I will also explain you why. And so we see that there is a cyclic kind of a structure, the imidazolic ring, and this is also heterocyclic like the proline, that is proline. So CO-. And next we have the non-polar aromatic R group, that is phenylalanine, tyrosine and tryptophan. So phenylalanine has a phenyl ring, and in the tyrosine you just have an addition of OH group. So this is like a benzyl group, and this is like an OH addition, phenyl. And tyrosine is similar to serine and threonine because all the three are carrying the hydroxyl groups. And then you have the tryptophan. So tryptophan is having an indole ring. Tyrosine you call it as a phenol ring. So tyrosine is phenol, phenylalanine is phenyl, and tryptophan is indole ring. So in this indole ring is also heterocyclic. So total we have 3 heterocyclic rings. One is indole ring, the imidazole ring, and the pyrrolidine, which is in the proline.